Welcome to the next session on glucagon. So, glucagon is a hormone released as part of the pancreas and it is synthesized as a pre-pro-glucagon. The pre-pro-glucagon is converted to pro-glucagon by removing the leader sequence or the signal sequence, and the pro-glucagon is further post-translationally modified by convertases to different peptides where you can see two major fractions of peptide fragments that are formed. One is through the PSEK2 dominant, which is most commonly found in the pancreas. And in that you can see glucagon, glisantin related pancreatic peptide or GRPP, intervening peptide, and major pro-glucagon fragment. The other post-translational modification of the pro-glucagon can be seen in the brain and in the intestine, where the convertases 1 and 3 will convert pro-glucagon to different peptides, which include the GLP-1 and 2, oxyntomodulin, the intervening peptide 2, and the glisantin. So these are the different ways where glucagon can be post-transcriptionally modified. So in this figure, you can see the mechanism of action of glucagon release. So glucagon is stored in secretory vesicles that you can see over here, and the glucagon release is mediated by calcium concentrations inside the cell, particularly the alpha cell. So under low glucose concentrations, you have the glucose transporter or the GLUT1 on the cell surface of the alpha cells, and there are unknown sensors which can act in membrane depolarization. And now we know that the unknown sensors are actually the GLUT transporters itself, and the membrane depolarization will open the sodium channels as well as the calcium channels. The sodium channel will increase the sodium concentration inside the cell by the movement of sodium from its higher concentration outside the cell to its lower concentration into the cell. So this causes a membrane depolarization, leading to the activation of the calcium channels, particularly the L-type and the N-type calcium channels. As a result, the calcium concentration inside the cell increases. So one of the things that you have to understand in this process is this process is independent of potassium. It's completely independent of the potassium ATPs, as we have seen in the case of insulin. So the intracellular calcium concentrations will cause the exocytosis of the secretory vesicles which contain glucagon. So this is a condition for low glucose concentrations. And consider the situation of high glucose concentrations. You can see that the GLUT1 or the glucose transporter also activates the unknown sensor. Probably it is a glucose transporter itself. And there are several islet factors like zinc, insulin, gamma aminobutyric acid, somatostatin, and other nutrients which cause membrane repolarization. So a repolarization is the exact opposite of the islet factors zinc, insulin, gamma aminobutyric acid, and somatostatin as well as other nutrients cause membrane repolarization. So repolarization, as we know, is the exact opposite of depolarization. So in repolarization, there is no change or the activation of the sodium channels or the voltage gated calcium channels, so that there is no intracellular influx of calcium. As a result, there is no exocytosis of glucagon. Again, this process is a potassium independent process, right? So the hypoglycemic stimulation of glucagon secretion occurs through the membrane depolarization, which is independent of the potassium activity. So the calcium concentration inside the cell causes the release of glucagon from the cell. So this is the intracellular signaling based on or leading to release of glucagon. So in this figure, you can see that the potassium ATPase channel and the sodium glucose transporter can depolarize the cell, but the depolarization is in such a manner that the action potential is less compared to a normal system. So as a result, there is a reduced glucose secretion. There is glucose secretion, but it is reduced. In the second situation where you have the task 1 and the store operated channels, calcium channels, uh, the task 1 is actually a potassium channel and the store operated channels are the calcium channels. So in the presence of task 1 and SOC, it increases the repolarization of the cell so that the action potential frequency is decreased and the glucagon secretion is also decreased because there is an intracellular calcium concentration is becoming lower in this state. So the potassium channel and the SOC or the store operated calcium channels which will mobilize the endoplasmic reticulum calcium concentrations will cause a repolarization in the cell which decreases the action potential and therefore glucagon secretion. So in this figure you can see that the glucagon receptor binds to glucagon and the glucagon receptor is a G protein coupled receptor which can activate the phospholipase C system and the adenyl cyclase system. The adenyl cyclase system activates or converts ATP to cyclic AMP. The cyclic AMP activates protein kinase A and the protein kinase A activates cyclic AMP response element binding protein and it activates several gene expressions for several proteins. So the phospholipase C converts phosphatidylinositol diphosphate to inositol triphosphate, which increases the calcium concentration inside the cell. The increased calcium concentration and the protein kinase A or the activated protein kinase A will cause the synthesis or the activation of the glycogenolysis pathway, gluconeogenesis pathway, and it will inhibit the glycolytic and the glycogenetic pathways. So this is what is being explained over here. So this is regarding the functions of glucagon and you know that glucagon is antagonistic to insulin. So whatever insulin function is, glucagon is the exact opposite of the function in insulin. So one of the major functions that has been most commonly studied or studied the most is regarding the hepatic glucose production. So the hepatic glucose production is increased whereby the glycogenolysis is increased, gluconeogenesis is increased, hepatocellular survival and lipid oxidation is increased, whereas glycogenesis is decreased. So this is one of the functions of glucagon that has been studied extensively. There are several other functions which is indicated by the dotted lines which have not been studied as extensively, including the intestine with a lowering of its gut motility, The kidney with an increase in the rate of glomerular filtration and water reabsorption. The brain with increased glucose production and increased satiety. And the heart with increase in cardiac inotropic effect, glucose oxidation and glycolysis. The adipose tissue with increase in lipolysis. And probably there is the function of the glucagon in stimulating insulin secretion by the beta cells, and also autoregulation of the pancreas is also completed by the glucagon. So this is the figure where you can see that consider your system where you have had food, and when you have had food your blood sugar concentration increases, and when the blood sugar concentration increases it signals the pancreas to release insulin. So the insulin will make up or will stimulate the adipose tissues to take up the glucose so that it decreases the blood sugar concentration. And consider the system of exercise where your blood glucose concentration has gone low and your pancreatic alpha cells will synthesize glucagon. So the glucagon will act on the liver, which breaks down glycogen to glucose and provides increase in the blood sugar levels. So this is sort of a reciprocal cycle which happens regarding the blood sugar. So glucagon secretion by the alpha cells are inhibited by insulin and parasympathetic activity and activated by sympathetic activity, secretin, cholecystokinin activity, increased amino acid concentration and decreased glucose concentration. So the glucagon secretion in effect affects the fatty acids and ketones. That is, it increases the fatty acid and ketone concentrations, and there is inhibition of anabolism. So then you have the secretion of insulin and increased glycogenolysis and gluconeogenesis. So this was regarding the glucagon and its function.